Good afternoon and welcome. My name is Tim Poe. I'm Director of Telehealth with the UNC Cancer Network and the UNC Lineberger Comprehensive Cancer Center. Thank you so much for being here today. I know there's a lot going on in the state of North Carolina with the storm, and so if you were able to attend live, we really appreciate that. For those of you, of your colleagues not able to attend live today, we will of course have the recording, and we'll have that in our learning portal for credit as well. Just a few reminders before we get started. Any technical difficulties, please call us 919-445-1000. A reminder to those of you attending via Zoom that in order to receive credit for the live presentation, you do need to attend at least 50 minutes of the presentation. Our Zoom system tracks that, so please be sure. We hope you're here for the whole hour, but a minimum of 50 minutes in order to be able to award that credit. We'll use Poll Everywhere today for audience response, and I'll talk about that in a moment. And we're on the web at unccn.org. We're on Facebook, Twitter, YouTube, lots of places you can find us. Hopefully you've already received this email, but if not, we are doing our 2019 lecture planning survey right now through the end of next week. Please take just a few minutes we've made it under half the length of last year's. So just 9 questions, should only take a few minutes. www.unc, oh my gosh, and we've got a typo on there, so I'm going to fix that. This is real time. We're going to fix, and of course the mouse went out. This is exciting here, but we're going to fix this in real time. unccn.org, and that goes ahead and takes you to our /survey. So unccn.org/survey, that will take you to our Qualtrics survey. Just a few minutes to fill that out. That will help us to put together an even as good or better a series next year as we've had this year. So we really look forward to your feedback. I have noticed a few folks have gotten a bounce back because of security features on their email. If it does that, just email us, unccn@unc.edu, and we'll get you the direct link if you need that. It's just a little longer than we want to use here. All right, we will be using Poll Everywhere. 2 ways you can use this today. You can go to pollev.com/UNCCN. You'll see the questions in real time as they appear, and you'll see the answers. You'll see what your colleagues as well as yourself have answered on that. So that's an easy way to get there. That works on any browser, including a phone browser. You may also use your phone and text to the number 22333, the letters UNCCN. So you're texting to the number 22333 with the letters UNCCN. You do that one time, you'll get a little message back that says you've joined, and then you can go ahead and respond with the appropriate letters for the questions. And then please be thinking of questions for Dr. Armistead and sending those to us at the end of the presentation. We'll have a Q&A period then. So the first question, immunotherapy works by doing which of the following? A, suppresses the immune system. B, uses radiation to attack the immune system. C, uses electrical impulses to attack the immune system. Or D, prevents the cancer from suppressing the immune system. So you can send that to us, and we'll take a look in a minute and see what we've got. And without further ado, I want to welcome Dr. Armistead. Dr. Armistead, thank you so much for being here today.
Thanks so much, Tim.
So let's see what we know about you. MD, PhD at the UNC Lineberger Comprehensive Cancer Center and the UNC School of Medicine here at University of Chapel Hill, North Carolina, and focus on cancer and procedures, research, stem cell transplantation, leukemia, leukemia antigens, minor histocompatibility antigens, leukemia, myelodysplastic syndrome, and lymphoma.
That's correct. Yeah.
That's a broad variety.
It's my clinical focus is primarily in leukemias, and we perform stem cell transplants, which is sort of the original immunotherapy. So that's how I got into this field.
Gotcha. So, and can you tell us a little bit more about your career trajectory and what got you here?
So I grew up in Western North Carolina, and went to UNC where I met my wife in the 90s, and I stayed here through my MD PhD, and in 2002 went to Boston and then Houston for my post-MD training, and then my wife and I love this area so much UNC was looking for transplanters, and we were delighted to come back. And we've been back now since 2009, and my eldest daughter comes to Carolina, and I don't really see us ever wanting to leave.
No, I can't imagine why you would. Well, good. Thank you so much. Well, let's take a look at that question, see how they're doing. We seem to be trending here on D. What do you think?
So I think D is the correct answer. So immunotherapy is a whole combination of different strategies where we try to prevent cancer from suppressing the immune system and allowing our immune systems to overcome a tumor that's developing.
Very good. Well, this is a very timely topic, Immunotherapy 101, and we're so glad to have you here to present today. Thankful to our audience for being here today, and without further ado, let me pass the controls over to you. If you want to use the mouse as a pointer, you're certainly welcome to. And then just the right and left. Perfect. All right. Thank you.
Well, thanks everybody for logging on a somewhat busy week. As I said, my name is Paul Armistead, and I'm Associate Professor of Medicine here at UNC, and clinically my focus is on stem cell transplant. So briefly, I just want to go over the objectives for this lecture, and the lecture is going to take about 50 minutes to an hour. There are three main objectives I'm wanting to accomplish today. And the first is I want to at least give a brief overview as to how the human immune system is organized, what its major components are, then how they work together to coordinate an active immune response against pathogens that we see pretty much every day. Then, after a very 30,000-foot view of the immune system, we'll talk a little bit about how tumors develop methods to avoid our immune system. And after discussing that for a few minutes, I'll talk about a few of the immunotherapies that are currently in use clinically and how they seek to either boost our own innate, our own intrinsic immunity, or actually by delivering other immunotherapeutic drugs directly to tumors. So briefly, as a little bit of a background, we've had a pretty good understanding that the immune system can control cancer for a long time. This has been known for 30, 40 years in many different instances. And this table here is more of just a summary to sort of show that patients who have immune deficiencies are at increased risk of cancer. If you look on the left, there's a list of several quite common cancers. And what this table shows is a compilation of various studies that looked at the number of patients who developed these tumors who are immunosuppressed versus what you would expect to see in a regular healthy population. So if you look up at the top, non-melanoma skin cancer, in a population of immunosuppressed patients, they identified 127 cases. And if this was what you would predict to be a healthy population, you would only expect about 5 cases in the population. And so as you look through here and you look at the column on the right, you observe that there's a ratio of observed cancer cases that's far higher than what you would expect. And this goes through many different tumors, all the way down to breast cancer, where you actually see not much of a difference. But the take-home point from this table is that there are many tumors where being immunosuppressed increases your risk of developing cancer. Most of the patients in these studies were immunosuppressed because they'd undergone solid organ transplant or an immunosuppression, but there were other reports that came out from patients who had HIV or AIDS or had other forms of immunodeficiencies. So having shown that being immunosuppressed does increase one's risk of cancer, we should probably think about what are the major features of the immune system and how does the immune system normally work, and then how can we enhance these responses to try and fight tumors as they develop. So if you look at this rather busy slide, you can look at the top and see that there are two main components of the immune system, what we call cellular immunity and also humoral immunity. And we'll start at the cellular immunity aspects first. And so cellular immunity we break down into two groups, which we call innate immunity and adaptive immunity. And we're not going to focus much on innate immunity in this talk, but the innate immune system is probably evolutionarily our most primitive form of immunity, and the innate immune system helps us recognize general patterns or chemicals associated with pathogens that cause very common diseases. And so the innate immune system is our complement system. It helps us fight bacteria. It does have some cellular components like NK, natural killer cells or macrophages that again can just kill bacteria or other very common pathogens, but it also works in coordinating with other aspects of the immune system through inflammation and also the presentation of antigen or immune recognition molecules to stimulate other parts of our immune system. Another aspect of cellular immunity, something that we will focus a lot on, is actually what's called the adaptive immune system. Adaptive immune system primarily focuses on what are called T cells. We'll talk a little bit more about T cells because they're actually increasingly important in understanding how our immune system is able to fight cancer. So the adaptive immune system has two main groups of cells, the helper T cells and the cytotoxic T cells, and those are particularly important in killing certain target cells, particularly cancer in our discussion today. And then the helper T cells help regulate this entire immune system by secreting cytokines that can influence the interplay of other cellular messengers in the overall immune system. The third component of the immune system that we'll talk quite a bit about is actually humoral immunity. And humoral immunity actually talks primarily about antibodies, but antibodies actually are derived from cells. So B cells, which are another type of immune cell, can be stimulated by antigens with the help of these helper T cells. The B cells can become plasma cells can secrete antibodies, and antibodies are one of the hallmarks of how we, first of all, neutralize pathogens, as is shown in this slide, but also we use antibody-based therapies routinely in targeting cancer and developing various immunotherapeutics. So briefly, we're going to discuss some of these components of the immune system, and just as a bit of a refresher, I will talk in a little more detail about what exactly antibodies are, so when we think about these therapeutically, we'll have a better understanding of how these are engineered and how they work. So antibodies, as I said, are secreted by plasma cells. The actual antibody structure is composed of two large proteins called the heavy chains, and there are two heavy chains in this diagram. One is in blue and one is in yellow. Those heavy chains are linked, and this region is called the Fc region. The other components of the antibody has a heavy chain linked with what's called a light chain. There are two light chains shown here. The light chains are in red. And this complex between the light chains and the heavy chains creates what's called the FAB region. And the antibody, when it's secreted, it goes around and it binds with very high affinity to certain pathogens or targets. And the recognition is mediated by the FAB region, which is where these light and heavy variable chains are. The Fc region is what you can consider sort of the effector portion of this molecule. This is the portion that actually interacts with other parts of the immune system to help generate an immune response to clear a pathogen or, in some cases, clear a tumor. And antibodies do this in several different mechanisms. And so we'll look at this slide now, and then we'll come back to it a little bit later on. But in this example, you have an antibody down here, and the FAB portion, which is where my pointer is binding to a membrane antigen. And this is just a protein that's sitting on a target cell. Now, this target cell could be a bacteria. We're talking about cancer immunotherapy today, so you can imagine this target cell being a tumor. The FAB portion binds to the target, which is called an antigen, and then several different processes can occur. If you look up here, you can see the FAB region binding to the antigen, and the FC portion, the tail end of the antibody, binds what's called the C1 complex. This is part of the complement cascade. When the complement cascade is fully assembled, this actually punches holes in the membranes of target cells, and this can lead to the death of a target cell. This process is called complement-dependent cytotoxicity, or CDC, and this is commonly used in the normal functioning immune system. Antibodies can do other things. You can have the FAB region again bind to the antigen, and you can have the FC region bind to something called the FC receptor. And this sits on various cells, including some cells in the innate immune system, such as NK cells. And if the FC receptor binds the FC portion of an antibody, the cell will release cytolytic granules, and these contain proteins that can again create pores in a target cell and lead to the death of this target cell. This process is called antibody-dependent cellular cytotoxicity. Again, it's another way in which antibodies can lead to the death of a target cell. The third mechanism that I'll briefly mention is called antibody-dependent cellular phagocytosis. And if you remember back from a very early biology, phagocytosis is whereby the process of one cell eating up another cell. And what happens here is that an antibody binds its target through the FAB region. The FC region binds to the FC receptor, but this time, instead of secreting granules, the effector cell, in this case it's likely a macrophage, will just go ahead and engulf the target cell, essentially eat it, and kill the target cell. So having antibodies means that we can target various cells through many different pathways. And we'll come back to these as we start going into immunotherapy a little bit later on in this lecture. Having talked a little bit about B cells and antibodies, I want to talk again somewhat about T cells and the adaptive cellular immune system, because these are also the main components that we're going to discuss when we get into actual cancer immunotherapies. So to go into a little more detail about how T cells work is, first of all, on the previous slide I showed cytotoxic T cells and then helper T cells. Cytotoxic T cells express something, a protein called CD8, and it's sitting off the T cell. Helper T cells express a protein called CD4. So if you hear other lectures talking about CD4 cells, you can think of them as helpers. CD8 cells, you can think of those as killers or cytotoxic. The way a CD8 cytotoxic T cell works is that any cell in the body that has a nucleus can take up, in this diagram, this is a virus particle. This can be any protein. The protein is going to be digested to give us peptides, and the peptides bind to a molecule called MHC. For people who work in the stem cell transplant field, which is where I work, in human beings this is also called HLA, and there are some slides later on in this slide deck that use HLA, but you can think of HLA and MHC as essentially interchangeable. The peptides bind to this class 1 MHC and are presented on the surface. And if there is a T cell that has a T cell receptor that binds with very high affinity, then this T cell will secrete proteins. The classic ones are called granzyme and perforin. These punch holes in the membrane, the plasma membrane of the cell. The enzymes get in and actually induce cell death. And this is a way that we routinely clear viral infections. But we can also harness this method to actually eradicate cancer cells. Helper T cells work a little bit differently, and we won't talk about them quite as much, but the way a helper T cell works is that they interact primarily with professional antigen-presenting cells. These cells are called dendritic cells, monocytes, and macrophages. These in the initial slide are actually part of the innate immune system. They can also actually interact with B cells too. The way these antigen-presenting cells work is that they actually engulf and take up a particle. They digest the large protein or virus into little peptides that are now presented by class 2 MHC instead of class 1 MHC. This peptide protein complex is presented on the cell's surface. A CD4 T cell now interacts, but instead of killing the cell, the CD4 T cell actually secretes other cytokines. I've shown two cytokines that are commonly secreted, interferon gamma and interleukin 2. These cytokines actually cause other cells to be drawn to this immune site and further stimulate the immune system. So in order to have a functional immune system, you actually need both cytotoxic T cells and helper T cells. For those of you who are more familiar with in the infectious disease world, patients who have HIV or AIDS suffer from destruction of these CD4 T cells and are then prone to multiple opportunistic infections. So I've talked about the main actors of the immune system, at least in terms to what we'll discuss later, which is cancer immunotherapy, being antibodies and T cells. I want to talk a little bit about how our immune system normally regulates, because this plays an important role as to why it's so hard to develop an active immune response to human cancers. and I'm going to discuss a concept called immune tolerance. And the way tolerance, what tolerance does is it helps us generate an immune system that's very good at fighting foreign pathogens but doesn't target our own organs. The way this works is that this focuses primarily on T cells. Pro-T cells, or immature T cells, are made in the bone marrow, and they migrate into our thymus gland. Our thymus gland is located in the upper part of our chest. And these pro-T cells develop what are called T cell receptors. And these T cell receptors are developed in the thymus gland, and they're exposed to multiple antigens presented on MHC. And these antigens are called self-peptides or self-antigens, and these are just derived from the proteins that our bodies just naturally make. If any T cells bind with a very high affinity here, these T cells are killed. This means that we get rid of T cells that should react to us. But we will make T cells that don't recognize self-peptides. These T cells are going to leave our thymus gland, and they're going to sit around in our bloodstream and our lymph nodes, and if we're exposed to some kind of foreign pathogen that has a non-self-peptide presented, we can then elicit a robust T cell response. The T cells can proliferate, they can kill if they're CD8 T cells, or if they're CD4 T cells, they can recruit other immune cells to help fight off what's typically an infection. One additional feature that I should bring up is this notion of peripheral tolerance. So central tolerance is the primary way that we make sure that the T cells in our body don't attack us and do attack foreign pathogens. But we have sort of a backup measure which is called peripheral tolerance. And if some T cells that actually could recognize self-peptides or self-antigens make it out of the thymus and into the bloodstream, they can still be made ineffective. If a T cell happens to recognize a self-peptide or a self-antigen, but it is not appropriately stimulated, this T cell will become what's called anergic. We'll come back to this term a couple of times later in the lecture. Anergy means that the T cell can recognize an antigen, but it can't mount an effective response. Alternatively, if a T cell recognizes self-antigen and is chronically or is highly stimulated, it can undergo a process called activation-induced self-death and just be killed in the periphery, or it can undergo a process called T cell exhaustion, where the T cell recognizes the antigen, it just, again, is not able to elicit an immune response. Cancer cells are extremely good at creating a peripheral tolerance environment, which makes it harder for our own intrinsic T cells to mount a robust response. So, to conclude this one little portion of this lecture, briefly, our immune system has three main components. The innate immune system is a non-specific response to foreign pathogens. There is really no memory response, but it will respond to non-specific stimuli that are secreted by bacteria or viruses. We're not going to focus much on the innate immune system as it pertains to cancer therapy. The adaptive immune system has two main components. B cells and the humoral immune response produce antibodies, and then the adaptive cellular response is primarily T cells, and there are two main flavors of these T cells. There are the CD8 cytotoxic T cells that can kill cells that are presenting a foreign antigen, and there are the CD4 T cells that help sort of regulate how this immune response is managed. I believe now we're coming to the first polling question. And this question is, central tolerance through the thymus directly affects which of the following immune T cell populations? Possible answers are B cells, T cells, NK cells, dendritic cells, or E, all of the above. We'll give a few minutes, a few seconds for people to chime in with their answers.
Sounds good. So again, if you're texting in, just put in the letter that you feel is the appropriate answer, or you can just answer if you're in a web browser. And looks like two are of particular interest, and,
right.
and one is leading.
One is leading. Oh, yes, and we've sort of settled out with 2/3 saying T cells and 1/3 saying all of the above. So T T cells were actually called T cells because they were first discovered migrating to and out of the thymus.
Mm-hmm.
So the thymus gland itself really is important for T cell tolerance. The thymus gland actually does not do anything for B cells, NK cells, or dendritic cells. So the answer to this one is actually B, T cells.
All right. Well, congratulations to our audience. They did well.
Yes, it is. So moving along, we'll talk a little bit about how does our immune system try to target cancer. And so something to remember is that for human history, infectious diseases have been much more lethal to human beings than cancer. As a result, our immune system is extremely well tuned to recognize and attack non-self-infectious agents. These can be bacteria, they can be parasites, they can be viruses, they can be molds. And we're able to fight this very well. Our innate immune system helps us develop inflammation towards chemicals made by bacteria or viruses. And then we have both humoral and cellular adaptive immune responses that can help fight non-self proteins from all these pathogens. The big difference with cancer is that cancer is significantly much more self than non-self, at least when you compare this to viruses or bacteria. And as a result, our immune system has a much harder time finding very good targets. This is a figure taken from a paper that was published in Science in 2013. On the left, it shows sort of schematically all the different tumors that were sampled for this paper. But I'll draw your attention to the right. What the right shows is shows the number of mutations that were identified in these different tumors. And if you start thinking through the size of the human genome has about 3 billion nucleotide bases in each cell. And really about 30 million of those are bases that encode proteins and make functional proteins that our cells are required to perform their activities. And then you start looking at it, out of the 30 million bases per cell, the number of mutations is really quite low. This colorectal MSI group is a type of colon cancer where there's a known defect in the cell's ability to repair DNA damage. And you would expect there to be a lot of mutations. And in this extremely highly mutated type of cancer, we see less than 1,000 mutations. And this is out of 30 million bases. Other tumors that are associated with being exposed to mutagens, so lung cancer, where the vast majority of lung cancers are a result of cigarette smoke with carcinogens in it, and melanoma, which is highly linked to exposure to UV radiation, we're now down only seeing about 100 or 200 mutations, again out of 30 million bases. Most other adult tumors are down at less than 100 mutations, and then the diseases that I treat, the leukemias, we're looking at maybe 10 to 20 mutations, and it only takes that many mutations to cause a tumor, but that means that there are very few genetic differences between a cancer cell and a normal blood cell or a normal colon cell or a normal lung cell. As such, it becomes very, very difficult for our immune systems to have good targets, and this is one of the biggest problems with cancer immunotherapy. So in addition to the fact that cancers don't have many mutations that make them very different from their source tissues, cancers have developed many other ways to evade our immune system. So this is actually one study reporting one patient, but it's extremely illustrative as to what tumors can do. In this patient had melanoma, which is a skin cancer, removed. 5 years after the tumor was removed, a new cancer showed up. The investigators looked at this cancer and saw that it expressed two proteins, and then they looked at immune cells called TILs, or tumor-infiltrating lymphocytes. These are the immune cells that are sitting are mixed in with the tumor. And they found these TILs recognized a peptide antigen derived from one of these proteins called MART1. Said, okay, this is great. The tumors are recognizing the antigen and they're trying to control it. This tumor was removed. Then 6 years later, another tumor in the same patient popped up in a different site, this time in a lymph node. The tumor was evaluated, and again the tumor is still making these proteins, but the tumor has now evolved so that it is no longer expressing an antigen from MART, it's expressing an antigen from tyrosinase, and now the T cells are having to recognize a different antigen. And this means that over the span of time, The tumor is still expressing the same proteins, but it is changing and expressing different antigens, which means that the immune system is constantly having to catch up and find new targets. And the only reason that this was able to be analyzed is that this patient had been living for 16 years and there were multiple biopsies being taken. In most patients, we're not able to perform these kind of detailed studies, but this serves as a very powerful illustration. In addition to tumors being able to control what kind of antigens are being presented, tumors can also affect how the T cells work. So I talked briefly about anergies. We were discussing how peripheral tolerance works. Well, tumors can induce peripheral tolerance. Tumors can secrete other cytokines that cause an upregulation of a protein called CTLA-4. We're going to get back to that a little bit later on. CTLA-4 can compete with other stimulatory molecules so that if an antigen-presenting cell recognizes a T cell and is presenting a cancer antigen, you get insufficient co-stimulation. As a result, this T cell is antigen-specific. It would recognize a cancer antigen, but it's not being appropriately primed or stimulated, and it can't mount a robust response. So tumors can induce anergy. Tumors can also induce something called T cell exhaustion, where the T cells are just chronically being exposed to a cancer antigen, and this chronic exposure leads to upregulation of other surface proteins on the T cell. Some of these proteins are now being targeted therapeutically, such as PD-1, and again, CTLA-4 shows up here, because these T cells now, even though they recognize the antigen, are now in a state of exhaustion and are not able to function as adequate effectors. In addition, there's a very crude method where tumor cells can actually eliminate T cells, where if a cancer cell expresses a protein called Fas ligand can interact with a protein called Fas on T cells and actually just directly kill the T cells. So these are three mechanisms where cancers can actually just affect T cell function. Then we've talked about how cancers can affect the types of antigens that they present, how tumors can actually directly affect how T cells work. Cancer cells actually create what's called a tumor microenvironment. When a tumor is resected, it's not just a compilation of cancer cells. There are other cells mixed in with that tumor. And by secreting various cytokines, cancer cells can actually cause other immunosuppressive cells to come into the tumor. These can be called tolerogenic dendritic cells. Tolerogenic dendritic cells actually can make cytotoxic T cells have less effector function. They can cause another type of immunosuppressive cell called regulatory T cells, or Tregs, to come in and again suppress T cell function. And there's another group of cells called myeloid-derived suppressor cells, which again are immunosuppressive. So these cells can be brought into a tumor, and that will further suppress any active T cell function. In addition to that, tumors have an aberrant blood vasculature, so a tumor is typically hypoxic and it tends to be more acidotic than a normal organ. And a good oxygen concentration and appropriate pH is necessary for T cell function. So the actual environment of the tumor itself will actually greatly impair T cell function or effector T cell function in many different tumors. And so after that sort of quick overview, the next polling question is, cancers can evade immune responses through which of the following mechanisms? There are 5 answers. Answer A, directed cytotoxicity of anti-tumor T cells. Answer B, downregulation of cancer antigens. C, chronic antigen stimulation of T cells leading to T cell exhaustion. And D, recruitment of immunosuppressive cells to the tumor environment. It seems like there's a consensus that came about pretty quickly on that.
I think everyone was listening carefully there.
And no, that is the correct answer. I think really the take-home point is that when we start thinking about immunotherapy, you hear about immunotherapy on the news, we're developing therapies that may affect one of these mechanisms of a tumor cell evading the immune system. But tumors have many, many different mechanisms. So if we're to create robust immunotherapies, we have to think about all of these different mechanisms.
All right, well done.
All right. So, quick summary of tumor immune evasion. As everybody answered, cancer cells are genetically very similar to normal cells, which means that if we're going to come up with a therapy, there are going to be very few good immune targets for targeting cancer. Cancer cells can actively target our own immune cells. They can cause energy and T cell exhaustion. Now, these are normal processes, but we need to be part of our normal peripheral tolerance, but cancer cells can enhance that for their own purposes. Cancer cells can also induce T cell death directly through this fast ligand interaction. Then finally, tumors don't just exist as nests of cancer cells. There's a whole microenvironment that develops with cells that come into that microenvironment that impede our immune cells' function. This is caused by hypoxia and impaired blood flow, an acidotic environment, and then the recruitment of other immunosuppressive cells. So, the last part of this talk, we'll go over different kinds of immunotherapeutics and just give people an idea of the kinds of immunotherapies that are currently being used, how they work, and maybe some of their relative strengths and weaknesses. So first of all, a way to think about immunotherapeutics is that you can have something called passive immunotherapy or you can have active immunotherapy. Passive immunotherapy is where you administer some kind of an immunotherapy agent to actually target the specific disease. This is a little different from active immunotherapy. Active immunotherapy is more when we give an agent designed to enhance the patient's immune response, and we're banking on the patient's immune response to actually eradicate the disease. And I've got lists of both passive and active immunotherapeutics that are in various stages of development. For this talk, I'm going to focus on these 4 immunotherapeutics because these are the ones that really we see most active in clinical practice. So for passive immunotherapeutics, we'll talk about anti-tumor antibodies. We'll also talk about CAR-modified lymphocytes, CAR-modified T cells. Then for the active immunotherapeutics, we're going to talk about what are called checkpoint inhibitors. And these are actually anti-CTLA-4, anti-PD-1, and anti-PD-L1 antibodies. So, targeting of cancer surface proteins. There are many tumors that overexpress proteins on their plasma membranes. Now, these proteins are typically not cancer specific. They're not a result of a mutation. This is a normal protein that just happens to exist at a high level on the plasma membrane of a cancer cell. So these have been targeted for many years and have greatly improved clinical outcomes. When these targeting strategies are developed, you do have to worry about targeting normal tissues because, as I said, these are not cancer specific. But the best immunotherapeutics directed against overexpressed cancer surface proteins are antibodies. And again, going back to why antibodies would be useful, this is a slide from earlier. There are 3 different mechanisms that are here. And one could imagine if you create an antibody that attacks a protein that's highly expressed on a tumor cell, you may be able to elicit either CDC, complement-dependent cytotoxicity, or ADCC, antibody-dependent cellular cytotoxicity, or even ADCP, which is antibody-dependent cellular phagocytosis, where any of these mechanisms could lead to the destruction of a target cell, this target cell being cancer. And just to fill things out, ADCC would typically be mediated by NK cells, and ADCP could be mediated by macrophages, and these are the components of the innate immune system. So antibodies could actually serve as a link between our humoral immune system and our innate immune system and give us many different ways to try to kill tumor cells. And this has been very, very effective. There are many studies I could show. I picked this study because it's just a very clean and striking example of what an antibody can do for treating patients with a common disease. So diffuse large B-cell lymphoma is the most common lymphoma in human beings. These cells express a protein called CD20, and it's highly expressed in diffuse large B-cell lymphomas, but it's present on other B cells. In this study, investigators in Western Canada looked at a large population of patients in that area and then asked a simple question. What was the survival for patients who were treated with standard chemotherapy, the diffuse large B-cell lymphoma patients treated with standard chemotherapy before the development of an anti-CD20 antibody called Brentuximab? And the 4 to 5 year survival was about 50%. And when I was in medical school, this was sort of the standard answer that we were given. Around 2000, Rituximab, or Rituxan, was developed, which targets CD20, and it was added to conventional chemotherapy. In this population in Western Canada, after Rituxan was added, the 3-year survival went up to about 75%, which is a striking difference in overall survival simply with the addition of this one antibody. As such, clinical outcomes for this relatively common lymphoma are much better now simply with the addition of this one antibody. These types of studies have been done with many different antibodies, but I think it's important to realize that conventional antibodies targeting cell surface proteins can have a dramatic effect on long-term survival. Now, since the development of their antibodies, we've come up with cleverer ways to try to improve antibody efficacy. One strategy has been called antibody-drug conjugates, where you have a conventional antibody, and to that antibody, a somewhat complex chemical structure is added to it. And I know that these labels are somewhat faint, so I've added cytotoxic agent here. This drug and this particular conjugate is a drug called MMAE, and this is a highly potent cytotoxic agent. You can't give this drug just by itself because it's too toxic to normal tissues. But if this agent is bound to an antibody through this long construct, you can then administer this entire drug. The FAB portion binds to the protein on the surface of the tumor. This antibody-drug conjugate actually gets taken up by the cancer cell, and the cancer cell's endogenous enzymes cleave at this drug cleavage site and now release the cytotoxic agent at high concentrations into the cancer cell, killing it. And this has been done in several contexts. The best known context is a drug called Brentuximab vedotin. Its brand name is Etcetras, and this is used to treat Hodgkin's lymphoma, which has a molecule called CD30 sitting on its surface. In the study on the left, patients who had Hodgkin's lymphoma that had come back after a stem cell transplant were either randomized to receive a placebo or Brentuximab for a year. And on the left, you can see progression-free survival, and the progression-free survival in the patients who received Brentuximab after a transplant was about 5 to 10% improved compared to patients receiving placebo. So again, it's an improvement, but when you think back to the Rituximab story, the difference is not quite as large. Brentuximab has most recently been reported in a study that came out of the New England Journal of Medicine earlier this year in combination with standard chemotherapy for treatment of patients with Hodgkin's lymphoma up front. So patients who are diagnosed with Hodgkin's lymphoma in the United States are typically treated with a regimen called ABVD. The problem with this regimen is that the B is a drug called bleomycin that has quite a lot of lung toxicity. This study looked at substituting out the B and the bleomycin and adding this A, which is the Etcetras or the Brentuximab. And again, looking at progression-free survival was again about 5% better in patients who received the Etcetras, this additional A, instead of receiving bleomycin. So progression-free survival was better. You would have to do a study for a much longer time period to see if there's an improvement in overall survival. But because bleomycin has lung toxicity, this is quite a reasonable approach. We can further modify antibodies and come up with something called bispecific antibodies. And these are even more engineered than the antibody-drug conjugates. The way a bispecific antibody works is that the light chain and heavy chain, FAB portion, is chemically linked to create what's called an SCFV. That's a single chain because it's one chain, variable region. And this is done for two antibodies, hence the reason bispecific. And the bispecific antibody we're going to talk about is a drug called Blinatumomab. And this antibody has, this bispecific antibody recognizes CD3 and CD19. Again, we talked about CD19 is another molecule that's expressed on a lot of lymphoma cells. And so this conjugate that looks very different from what an antibody is now infused. The CD19 binds to the CD19 that's expressed on a tumor cell. Again, this is often seen in B-cell lymphomas and leukemias, and the CD3 binds to normal T cells. This actually pulls normal T cells close to the tumor, activating them, and then the T cells, like conventional CD8 T cells, secrete granzyme and perforin and can actually kill tumor cells. And so this is being developed in a drug called blinatumomab, and it's used to treat a disease called acute lymphoblastic leukemia, or specifically B-cell acute lymphoblastic leukemia. This study that was published in the New England Journal of Medicine in 2017 looked at patients with extremely aggressive ALL. This is ALL that is now relapsed and is refractory to other therapies. These patients were randomly assigned to either blinatumomab or chemotherapy. The overall survival here was improved to about median overall survival about 7 months versus 4 months with conventional chemotherapy. And again, you can see that survival is still quite poor. So it's an advanced therapy that does improve survival, but is by no means a home run. The one thing that this therapy does, however, is it does actually lead to a third of patients going into a complete remission with full hematologic recovery. These are patients who could go on to an allogeneic stem cell transplant, which could have a chance of curing them. This is substantially better than what we see in a chemotherapy group, but again, this is an advanced therapeutic that is not likely to be highly curative. So, quick summary of antibodies. Antibodies target cell surface proteins. These surface proteins are not cancer specific. These therapeutics have improved clinical outcomes. Some of the bare antibody therapies, such as Rituximab for diffuse large B-cell lymphoma, have greatly improved our survival rates. Antibody-drug conjugates and bispecific antibodies are clinically used now, and they do improve outcomes. However, the magnitude of difference is not quite as not as good as we would have hoped. I'm going to briefly move to what's considered a very hot topic in this field, which is chimeric antigen receptor T cells. Now, going back to the discussion of CD8 T cells, the first part of this lecture, I discussed how a T cell has a T cell receptor, and it has an alpha chain and a beta chain, and it recognizes a peptide antigen presented by HLA. Now remember, in the first slide it talked about MHC, but in human beings this would be HLA. And with this binding and recognition, this T cell can then kill a target cell. Chimeric antigen receptor is actually a genetically engineered receptor where an SCFV, very similar to what I described in the bispecific antibodies, is now linked to a co-stimulatory domain. This molecule CD28 shows up in a couple of earlier slides, and then the T cell signaling complex, which is this thing called CD3 zeta. And this chimeric antigen receptor can be genetically engineered into T cells to recognize a tumor antigen, and now you get T cell mediated killing with the specificity essentially of an antibody. And CD19 keeps coming up, and we should discuss why CD19 is going to be used, because the next few slides are targeting CD19. So blinatumomab in the bispecific antibody also targets CD19, and it's because CD19 is expressed in these cells, which are B cells, and you could conceivably eradicate B cells. You still have T cells that are present, because there's virtually no expression of CD19 here, and all of the myeloid cells, the monocytes, the myelocytes, promyelocytes, the neutrophils, all of these other cells in our myeloid lineage do not express CD19. So if you come up with an effective therapy to target CD19 that is expressed by the tumor, you can get rid of the tumor, you can get rid of B cells, but you can preserve the rest of the immune system. The way CAR cells work is that blood is obtained from a patient, the T cells are selected, the T cells are then infected with a virus that contains the gene to actually express this chimeric antigen receptor. The T cells are expanded, they are then put into a bag. The patient receives chemo to get rid of a large percentage of their own T cells, and then these chimeric antigen receptor modified T cells are infused. This study came out in 2017 to show the effects of a commercially available product called Yescarta. This is to treat, again, aggressive relapse lymphomas. You can see there's an 82% response rate. 50% of patients developed a complete response. And what was striking is that the patients who developed a complete response, 70% of those patients maintained their complete response for 2 years with really no evidence of continued relapse, indicating that there's a very good likelihood that these patients were cured. Another CAR T cell, again targeting CD19, that's commercially available, is called Kymriah. This study was looking at a pediatric population. The ages were from the age of 3 to 23 who had relapsed and refractory ALL, the same type of disease as being treated with blinatumomab. Patients again received the CAR T cells, and you can see that the event-free survival at 1 year was about 50%. Now the overall survival you see is dropping off, and what is likely happening is that as patients relapsed, they would be further treated, but unfortunately these patients ended up succumbing from their disease. But it looks as if after about 1 year, the incidence of relapse is dropping off. So there's a very good chance in these patients with extremely aggressive leukemia that the cure rate is going to be in the order of 50% with these long-term studies. However, we do not have those data back. As I said, this study was published in 2017. So CAR T cells are toxic, and I think particularly nurses working in units that are going to be receiving patients with CAR T cells need to be aware of the quite high levels of toxicity. There's a condition called cytokine release syndrome, or CRS, which can actually look like sepsis with ARDS, where patients get high fevers, they can become hypotensive, they can actually develop an oxygen requirement, and we have grading for this therapy. And it's important to realize that close to half of patients who receive this type of therapy are going to get a grade 3 CRS, which can require very aggressive therapy. The kind of therapy that is used for management of cytokine release syndrome includes antipyretics for fever. Because these patients are presenting with fever and hypotension, they do need to be treated appropriately for potential sepsis with fluids and antibiotics. One intervention that is unique to the management of cytokine release syndrome is the administration of agents targeting interleukin-6. Interleukin-6 is an inflammatory cytokine that mediates the fevers and the hypotension. There's a drug called Toxicity that can be given to counteract interleukin-6, and that is also given for treatment of patients with cytokine release syndrome if it reaches what's considered to be grade 2. In addition, because these patients are hypotensive, they may need to be treated with vasopressors, and in severe cases, steroids can be used, although the evidence to support that is more limited. In addition to cytokine release syndrome, there can be neurotoxicity from CAR T cells. The cytokine release syndrome typically develops about 4 to 7 days after T cell infusion. Neurotoxicity can occur at the same time as cytokine release syndrome, but oftentimes it occurs later, about 1 to 2 weeks after infusion of CAR T cells. The symptoms can range from mild confusion all the way up to coma, and again there are various grading scales that are used to categorize the neurotoxicity. Neurotoxicity management is actually somewhat more complicated in that we do not have highly effective therapies. If patients develop neurotoxicity, steroids can be given. If the patients have cytokine release syndrome in addition, tocilizumab, the anti-IL-6 antibody, can be given, but we do not expect tocilizumab to work in and of itself for managing neurotoxicity. So as a result, patients who have neurotoxicity attributable to CAR T cells but do not have cytokine release syndrome should not receive tocilizumab. It should only be given in patients with concurrent cytokine release syndrome. And moving on to the final polling question. Which of the following are true of all antibody-derived immunotherapeutics? A, they are cancer-specific. B, they kill tumor cells by complement-dependent cytotoxicity. C, they target extracellular membrane-bound proteins. D, on target, off-tumor side effects are rare. Or E, none of the above. Right, we're somewhat split. I'm going to have better diversity of answers next time. So we're settling out maybe at 3/4, just saying that they target extracellular membrane-bound proteins, and then E is none of the above. So the correct answer is C. Antibodies always recognize what's being presented on the cell surface. Now, sometimes they can bind to the cell surface, and because of their Fc receptor, they can cause ADCC, CDC, or antibody-dependent cellular phagocytosis, but they can also be internalized, as in the case of Brentuximab, with the release of a cytotoxic payload, but they always at least recognize an extracellular membrane-bound protein. Okay. All right. So the last topic, so briefly, to go over the toxicities that we see, the main points from this slide are that both Kymriah and Yescarta, which are commercially available, and UNC has other CAR T cells under development here, are associated with substantial degrees of cytokine release syndrome and relatively high rates of neurotoxicity, with cytokine release syndrome being seen up to about 50% of patients, and neurotoxicity maybe up to 30% of patients. In summary, CAR T cells, they can provide long-term complete responses to patients with aggressive hematologic malignancies. Results in solid tumors have not been as encouraging. Unfortunately, for CAR T cells, there are very few good targets. You need to have a highly expressed surface protein that's expressed on cancer cells, that's not expressed in other tumors, or if it is expressed, these other tissues are not required for normal human physiology, and that's a very hard thing to find. This is a personalized therapy that's very expensive, and there's also significant toxicity with these therapies. Briefly, at the last few slides, we'll discuss the immune checkpoint inhibitors. These are therapies that are considered active immunotherapy because they don't so much target the tumor, they enhance the intrinsic ability of the patient's immune system to fight the tumor. Something that's important to realize is that checkpoint inhibitors can work in two areas. There's what's called a priming phase and an effector phase, and we have drugs that work on both of these. The priming phase is where a T cell interacts with an antigen-presenting cell, in this case a dendritic cell. The T cell receptor recognizes the antigen, it could be a cancer antigen with its MHC, in humans it would be an HLA, and in normal cases there are activation signals. However, there could be, for some reason, upregulation in the T cell of CTLA-4. And this CTLA-4 can interact with the B7 activation signal, and this would lead to cells essentially becoming anergic. However, if you give an antibody to block CTLA-4, you will improve T cell priming. Sorry,
I'm sorry, I don't know what happened there. We'll go ahead and see if we can take care of that. Looks like we have somebody else who tried to share some content, and we're going to see if we can get our content back.
We
had it for a moment for our team and back. If you can take a look and see, there we go. Looked like, again, somebody else was trying to share content, but we are back in the game.
Okay. So T cell priming, this would enable you, if you could prevent CTLA-4, you could cause these T cells, instead of becoming anergic, to actually become activated and have a strong effector function. The effector phase could be a T cell interacting with a cancer cell, where again the T cell receptor recognizes the target, but now if the T cell is being chronically stimulated, it may start increasing its expression of PD-1, which will make its effector function, its ability to kill, decreased. Alternatively, the tumor cell can actually increase the amount of PD-L1 it's making. If there's a lot of PD-L1, it will interact with the PD-1 and again impact the ability of the T cell to actually have effective killing. So as such, we could, and we are, targeting all of these different molecules to try and enhance different T cell function. The first drug that was developed was anti-CTLA-4 or Ipilimumab, and this was developed for the treatment of advanced melanoma, where patients had advanced melanoma that was surgically resected, but then they were either given a placebo or Ipilimumab, and then their recurrence rate was measured over the course of this study. We're up to 7 years. And the recurrence rate at 5 years was about 40%. Patients who did not have recurrence was about 40%, whereas patients who did not have recurrence who received placebo was about 30%. So there's a 10% improvement in the risk of the disease returning, and this translated into about a 10% improvement in overall survival with this initial therapy. And this was published in 2016. Now this was targeting, again, this is the priming phase. Then you can also look at the effector phase, and this is using an antibody called nivolumab, which targets PD-1. Remember, this is the effector phase. And again, patients with high-risk melanoma that have been surgically resected but are at risk of the disease coming back either receive nivolumab or ipilimumab. Again, this is looking at relapse-free survival, and again, the difference in relapse-free survival is approximating 15% in this study, indicating that nivolumab is even more effective than ipilimumab, bearing in mind that the actual duration of time in this study was shorter than the previous study I showed. You can imagine that if one drug works well and the other drug works better, the combination is better yet. This study was published last year, and this was looking at patients who had advanced melanoma that was to the point that it could not be surgically resected. So these patients had active disease and were now just treated with either one drug or the other drug or both drugs combined. And now looking at 3 years, 40% of patients showed no evidence of disease progression. And if you look at overall survival, these are patients with extremely aggressive melanoma, and there's a 60% overall survival result out at 3 years, again, better than either drug individually. The prior slides looking at nivolumab looked at anti-PD-1. There's a recent paper that came out earlier this year that's now looking at anti-PD-L1. PD-L1 is that molecule that can be expressed by tumors that can also downregulate the effector function of T cells. This drug is atezolizumab, and it was combined with a conventional chemotherapy regimen looking at patients who have metastatic lung cancer. And the chemo regimen was bevacizumab plus carboplatin paclitaxel, BCP, which is a common regimen. And again, progression-free survival was looked at 12 months. And you can see the progression-free survival was roughly double at 12 months, at 36% versus 18%. And you can see this here with also a median improvement in overall survival of 8.3 versus 6.8 months. Again, this is an improvement, but this is by no means curative at this point. If you look here, overall survival, when you're getting out to 3 years, is essentially the same, but the median overall survival is like 19 months versus 14 months. Just again, it's a 5% improvement in median survival, which is good, but there's definitely a long ways to go in the improvement of immunotherapy for lung cancer. Something to note is that these drugs do have toxicity. Going back to the nivolumab ipilimumab study, however, you can see that the percentage of patients with adverse events was about 1 in 4 who received nivolumab versus 50% of patients who received ipilimumab. And the major toxicities, diarrhea from colitis is common in ipilimumab. We don't see that in the anti-PD-1 therapies. Other autoimmune conditions, so hypothyroidism or hypophysitis, which is immune targeting of the pituitary gland, is again somewhat more common in the anti-CTLA-4 antibodies than the anti-PD-1 antibodies. Last slide is what the checkpoint inhibitors are doing is we believe that they are actually targeting what are called neoantigens. These checkpoint inhibitors seem to work best in targeting cells that produce a lot of these non-self peptides that come about from either mutations or gene fusions or alternative splicing that is cancer specific. Because there is no central tolerance, the checkpoint inhibitors are targeting actually the peripheral tolerance that is being induced by tumors. Again, this is illustrated by the fact that if you look at where most of our therapies are being directed, they're being directed at these types of tumors, which is lung cancer, melanoma, and then colon cancer with these high numbers of mutations. So in summary, checkpoint inhibitors, they do enable cancer antigen-specific T cell targets to target tumors, which means that they're active immunotherapies. They do work on different aspects of the immune checkpoint, which provides us with an intellectual reason for combining some of these therapies. And there are additional checkpoint inhibitors that are currently under clinical development. I am sorry for going over for a few minutes, but obviously I'll be happy to take any questions.
Thank you so much, and we'll go ahead and let me progress to that slide. So we don't have a lot of time for questions, but I hope you will try to get a couple in now. And if you want to email us questions at unccn@unc.edu, we can go ahead and take those after and forward those to Dr. Armistead. As we're waiting for just a minute for any questions to come in, with the CAR T-cell therapy, what is the viability for that to be repeated? If that's initially successful, you see a good response for a few years, can that be repeated, or is that unusual?
I think it's unusual in that if patients have a response and it's a strong response and they eradicate their disease, it's late relapses are uncommon.
Okay.
If the disease is going to come back, it typically declares itself quickly.
Okay.
And so I'm trying to think of instances. If it were to come back late, you would want to see what's happening in the tumor, because the tumor may have stopped expressing CD19, for instance, and all of a sudden your CAR T cells aren't recognizing the antigen. So that happens. Our biggest problem is usually the CAR T cells don't persist, and then there's relapse that comes back quickly. And that's been one of the major problems in this therapy. However, if the disease goes into a complete remission, but you're not able to detect disease and patients are going out for a long duration of time, the risk of relapse drops dramatically.
Okay. All right. Well, thank you. We don't have any questions in yet, and I know that everyone's busy and needs to be getting to other meetings and functions. So let me just quickly wrap up. We want to say thank you to the North Carolina General Assembly for their generous support of UNC Lineberger Comprehensive Cancer Center and the UCRF grant. So we thank them. We want to thank Lineberger, UNC Lineberger Comprehensive Cancer Center. We want to thank our team for making each of these possible. So Mary King, Veneranda Obore, and John Powell, thank you all. Coming up, Medical and Surgical Oncology Lecture, 2 weeks from today, September 26th, Optune and newly diagnosed glioblastoma with Dr. Kagi, and we're excited about this. There was just some nice press on WRAL about the Optune, and so we hope you'll have a chance to look at that between now and the time of that presentation, and then he will bring the Optune in for demonstration at that time. And then our next RN and Allied Health Lecture, Understanding Oncology Drug Interactions, with Dr. Morgan, so we'll look forward to that as well. Our self-paced courses are there, and you can find out all about that on our site. 2 new ones are available, Novel Molecular Targeted Therapies and Biomarkers in Advanced Colorectal Cancer with Dr. Lee, and Palliative Care Opportunities with Nicole Sartor. So we hope that you'll, if you haven't been able to attend the live courses, that you can take the self-paced online courses on our site. And please visit that survey. There's a typo there, www.unccn.org/survey. And again, just let us know if you have any trouble with that correct link. All right. Thank you all for participating. Dr. Armistead, thank you so much for being here today. Until next time, stay safe. We hope that you all weather the storm successfully and are doing all that you can to prepare for that. And we look forward to seeing you in a few weeks. Thanks so much.